So remember the key with this is phagocytosis and inflammation. So we talked about hematopoiesis, where we get our cells, white blood cells. We talked about this phagocytic process in which your pathogen ends up in this phagolysosome, and we get degradation. I left you with chronic granulomatous disease, where your white blood cells can phagocytize the pathogen, but there's no oxidative burst. And then Shediac Higashi, where the white blood cells don't even have the ability to phagocytize. And if we can't get those pathogens in and break them down, I should say, then we always have problems. Now recognize this phagocytic process is the start of one of our specific immune responses. Even though it's not completely separate, it does its own thing. But once we end up getting that pathogen degraded, this cell differentiates into a completely different cell called an antigen-presenting cell. And it goes on and interacts with the specific immune responses, and we'll come back to that. So with that being said, we need to deal with the second part of our innate nonspecific response, inflammation. And inflammation is kind of a large, generalized term. And it has a few different things that are associated with it, of course. And it can come from both host factors as well as pathogen factors. So if these are coming from host factors, we call these, or I should say, pathogen factors. Exogenous pyrogens. And if it's coming from the host, these are endogenous. And you're going to hear this term exogenous and endogenous kind of over and over, just meaning outside of the cell or from within inside of the cell. And so one of these host factors that kind of increases the inflammation response are these things called cytokines. These are inflammation, or I should say immune response communication molecules, interleukins, growth factors, and a couple of others that we'll go through here in a little bit. One of the big things we see with these pyrogenic factors, of course, is fever. And so we think of fever as being stimulated by these pyrogens. And as I said, you can have both host and pathogen-encoded pyrogens. So we think of a fever as anything above normal temp for humans, which is somewhere between 97.2 and 99.5 degrees Fahrenheit, depending on your physiology, everything that's going on. So everything that's 100 degrees and above is considered a true fever. When it's low, right at the 100-degree mark, we kind of call that a low-grade chronic. And as it gets higher, we think of it as being more acute. So this inflammation response in itself basically is an augmentation process. It augments a lot of the other immune responses that are out there and kind of gets everything prepped for the infection. And there are a few different defense systems it augments. We won't go into all of them, but we'll deal with a few of them. First of all, stimulates the activity of white blood cells. And so they end up circulating in higher numbers, they're more active, they have the ability to phagocytize better. And of course, the overall goal here is to destroy pathogen and eventually display this pathogen to other immune systems. Now, fever in itself and these other inflammation responses we see, but fever particularly reduces free iron in plasma. And iron is key for a ton of different enzymes, of course, for your red blood cells, heme. But when we reduce the free iron in the plasma specifically, we're basically decreasing pathogen replication because their enzymes aren't getting the iron they need in order to work. It decreases toxin production. Increases other immune responses, specifically that rely upon iron as an indicator. Still talking about these augmentations. It stimulates the production of these immune hormones called cytokines. And these immune hormones are really there to signal. So they're big in signaling from one organism to another. 2 L's of signaling? 1 L? 2, it doesn't look right.
1.
What you need is the spell check board.
1.
You write on it and it just like automatically, it's 1.
Yeah.
Auto-correcting board, I think that's called a smart board, isn't it?
Yeah.
Star board.
But half of those things don't work either.
Right. And there are about 6 to 10 different classes of cytokines. So examples of these would be what we call interleukins. And you have interleukins 1 through, I think, the teens. But interleukin 1 plays a large role in signaling between immune responses. Some of these also can be endogenous pyrogens. And so these cytokines can act to stimulate the inflammation response. And then, of course, because we're at a higher temperature, it slows and kills the pathogen over a period of time. Bacteria really can't handle anything over about 104 for very long. So when you have an acute fever at about 106 to 108, of course you're close to dying, but you're also killing off the pathogen itself. Mixed bag, isn't it? And you'll find that's kind of how it is with the host immune systems. Many times they are just as responsible for problems you see as the pathogens themselves. So with our inflammation response, we can break it down into 3 major events. So we can go through vasodilation, squeeze things down a little bit. We increase the blood flow, the pressure moving around. We also, at the same time, increase the permeability of capillaries. We increase the plasma and the proteins and whatnot that are associated with that. Remember, when we think about the humoral system, this is our blood system, right? It really consists of 2 interlinked systems. Our blood system, our veins, our vessels, our arteries, whatever, and our lymphatic ducts, our lymphatic system. So your veins are leaky, okay? Plasma and proteins leak back and forth out of your vessels and veins, and this is what basically becomes your plasma. And then the last thing we see. We see accumulation of the white blood cells, usually at the site of infection. All of this together gives us an overall state in which we get help in localizing the infection. It stops spreading. So we keep it localized instead of becoming systemic or septic. We have the ability to neutralize pathogens and the toxins they produce. And we tend to see an increase in tissue repair, specifically of any of those cytotoxins that chew up and destroy our tissue. So when we take a look at a localized inflammation response, all of this kind of leads up to a couple of different things. We see them and know them quite well. Oh, I think I gave you guys a few bits of advice about what not to do before going to bed last time. Did anyone try the Pepto-Bismol? No?
No, I like my taste buds.
What? No, no. Remember, don't brush your teeth.
Yeah.
Take a couple Pepto-Bismol pills or some Pepto-Bismol and hold it in your mouth until you fall asleep. Just kind of let it drink down and you'll be good. You'll love it, especially if you have kids. Did you do it?
Well, I was sick last night, but my tongue turned like kind of black. It was so gross.
Ew.
Yeah, if you have kids, it'll turn their whole mouth black, and it doesn't come off their teeth or their tongue or something, no matter how hard they brush. So they go to school with this kind of gross mouth. So someone did do it, right?
Did you have the equivalent of the binocular strip with this class?
No, I haven't. So with our inflammation response, like I said, when we start to take a look at these close-up spots of these localized infections, what we end up getting is kind of the beginnings of what we call inflammatory exudate. So think of this as kind of all the liquids and stuff. So anytime you get a blister, that's a good example of this exudate, that liquid that's inside the blister that's trying to heal your skin. This is kind of what you see. So it's an accumulation of fluids, plasma proteins, different types of cells, cell debris, etc. And then eventually this inflammatory exudate starts to thicken up and turn a little yellowish and green. Really wonderful. And eventually turns into what we call purulent exudate. We know it as pus. So this is kind of that greenish, yellow goo, especially like if you had a sticker and it kind of festers and you push on it, you can see that purulent exudate come on out. It's a mixture of both living and dead cells, decaying or broken down organisms, so we get all sorts of cell debris that's in there, white blood cells, and other things as well, various factors. And it kind of basically turns into pus. So, something you shouldn't do before you go to bed, number 4, don't Google infected carbuncles, furuncles. Have you guys seen those? So it starts out as a boil. You know, we've all had like a big zit. It's a boil. It's really kind of subcutaneous. It's got a lot of pressure. Just normal kind of acne. That boil moves on to a carbuncle. Much deeper, much bigger, and eventually a furuncle. If you want to see some cool stuff, get on YouTube and look up draining a furuncle. Because this pus gets really stringy. Like you'll see videos of people stringing the pus out like feet and feet away. Or, you know, like these big, blossoming, gross cottage cheese furuncles exploding on someone's back. My kids love them. They just watch them all the time. Look at this one, it's a big exploding zit. It's like, no, that's disgusting. If you want to look at something, because these can be as big as like softballs. And they're really, really painful, especially
How do
for
they
my
get
kid.
that bad? Like,
What's that?
how do they get that bad? Are they like not cleaning themselves? Like,
No.
It
It's
just kinda
much more of an
happens?
infection. So, for instance, Propionibacterium, this natural organism that's on your skin that's really waxy, can produce it. If it gets subcutaneous, it gets in there and it starts to infect it. Otherwise, it's just this exudate that gets bigger and bigger and bigger. It stretches the skin. My dog had something like that. We call them abscesses in like animals, right? So she got a great big thorn in her side, didn't know it because she has so much hair. Turned into this big abscess. It's just the body through this purulent exudate is trying to surround it, degrade it, and get rid of it. Sometimes it doesn't work so well. With that, we get to move on to now the specific. And this is where things get a little crazy and a lot more molecular again. And then we'll pop back out of the molecular world. So we have these innate responses that are always turned on, non-specific. That's how I like to think of them. Always turned on, non-specific. Both these chemical and physical barriers. For your specific, these are based upon the specific pathogen that's infecting you. So, think of this as being pathogen specific. We break down this specific response into two different systems. The humoral, so everything that's hanging out in your blood and lymph. So those would be organisms moving around your body. And then we have the cellular response for those organisms that go intracellular, like viruses. Both of these systems, it's key that we have this recognition of self from non-self. And by non-self, we think of it as pathogens. Because what we don't want to do is spur an autoimmune response towards your tissues. Happens all the time. Lupus, Crohn's disease, ulcerative colitis, arthritis, many different ways that we get these autoimmune diseases kind of forced back upon ourselves. So what's key is that we have this quality control to make sure we don't have an immune response towards self, what we call self-antigens in this case. So now we have to kind of talk about what's going on. When we have the actual whole organisms, we call these guys pathogens, right? So this could be a virus, a bacteria, a fungus. But once it enters the white blood cell, we get that lysosome, or specifically that phagolysosome, breaking the pathogen down into little bits. That broken down pathogen the debris from this we end up calling antigens. And so your immune response can recognize both whole organisms or chunks of broken down organism, which we call antigens. Now, Usually this is cell debris. It can be lipopolysaccharides, it can be membranes, usually not nucleic acids, but carbohydrates, proteins, all of that, toxins. And we have specific attributes to these antigens. First and foremost, it has to be large enough to be recognized. So if the molecule is too small, we just don't recognize it as being formed or even being there at all. Then it has to have two different epitopes. And think of epitopes as two different three-dimensional surfaces. So for instance, if we take this over here and kind of blow it up, I have this chunk or protein, that's my debris. My immune system can recognize different three-dimensional parts of that antigen. So, in the future, I hate writing out antigen, so you're going to see it as AG quite a bit. This antigen can have three different shapes. So here's one example. Here's epitope number one shape of the protein that's completely distinct and different from the second one, epitope number 2. So it has to be large enough that two different surfaces can be recognized on it. Think of it that way. And of course, the big one, it's got to be able to stimulate an immune response. Many times, we'll go through this whole process. We'll break down a pathogen into these little antigens, and then we don't actually have an immune response to it. So it's key that we have this immune response. Now, these antigens are presented by antigen-presenting cells. And we call these APCs, antigen-presenting cells. We have a few different types of antigen-presenting cells. I'm not going to go through the whole litany of them, but I'll give you an example of some of them. So, antigen-presenting cells in the humoral system are these white blood cells that basically phagocytize and break these things up into their little antigenic epitopes. So white blood cells in the humoral system. Dendritic cells also play a pretty big part. In the cellular system, these are virus-infected cells or tumor cells, also dendritic cells. So there are two different pathways to display the antigens or epitopes. We display them one way through the humoral system, through these white blood cells, and we display them a completely second, different way in the cellular system. So one of the key players in all of this is this specific set of receptors, proteins, that have this ability to recognize self from non-self. And we call it the major histocompatibility complex, or the MHC. And it's made of a series of protein receptors. This is the name of it in our murine counterpart. Believe it or not, mice immunologically are closer to us than most other model animals, except for primates, of course. So a lot of what we find out about the immune system comes from these mice or murine model systems. And this is an example. The name, the major histocompatibility complex, comes from mice, but we use it interchangeably. In the humans, it's called the HLA system. They're identical. They're homologs of one another. You could take it out of the mice, put it into the human. It's basically the same functional thing. So this major histocompatibility unit consists of two different types of receptors. MHC class 1 type receptors and MHC class 2 receptors. So if we kind of show this membrane of our antigen presenting cell, here's our APC. In this case, when our MHC class 2, it consists of two identical transmembrane protein receptors. So I like to represent them as such. I have this transmembrane part, like that. And it's this MHC2, class 1 and class 2, that are actually physically presenting the antigen on the outside of the cell. So our MHC class 2 is going to basically end up interacting with its antigen. And on the other side, our MHC class 1, they're a little bit different. They're not completely identical. It has a very similar MHC class 2 type in this beta microglobulin. So it's not quite the same. They're a little bit different. And same thing, it interacts with the antigen. These guys recognize self from non-self. So if this antigen is self, it shuts down the whole process. So it's this kind of quality control or quality feedback. Same thing here. If this antigen is a self-antigen and it's being represented by MHC1, it basically kind of shuts down the whole system and says, wait, this isn't a pathogen. We don't want to stimulate this really strong immune response. So with that being said, let's talk about our first system. I'm doing okay on time here. And of course, I'm blowing this up very cartoonish in my Australopithecine stick figure type representations, but I think it'll get the point across. So our first specific system is the humoral system. You're going to see this for sure on your final. I can see an essay question saying sketch and describe the humoral response to an antigen. And I'm basically looking for this whole process. So, once again, this is in our blood and lymphatic system, right? So the cells that are involved, I always like to write those down so I can kind of think of these as I sketch it out. White blood cells, they're going to turn into antigen-presenting cells. They're going to interact with T helper cells. They do this via MHC2 molecules. We end up getting B cell differentiation into plasma cells. And from this, we get antibodies. So the antibodies are the main goal here. These antibodies are highly specific. We just don't make antibodies to many different things. And that antibody is going to be specific to the antigen being presented by that white blood cell. So I'm going to do this kind of in cartoonish form. Let's start with that white blood cell. We've been here before. This is what we talked about, phagocytosis. We get this active phagocytosis of our pathogen. During that phagocytosis, we end up forming this vacuole inside here called our phagolysosome. I just call it a lysosome for short. And of course, remember, in our phagolysosome, we're getting a decrease in pH, increase in proteinases, oxidative burst, and eventually we smash that thing up into bits. Basically, into those little antigenic pieces. So, this white blood cell, while this is happening, is going to have one vacuole, or phagolysosome, with our little bits of pathogen we just broke up. At the same time this is taking place, this white blood cell produces a secondary vacuole that has our MHC molecule, MHC2. These guys combine together, and this white blood cell now becomes an antigen-presenting cell. In that, it's going to have all of these MHC2 receptors on the outside of it, representing all these little bits of broken down pathogen or antigens. So all over this white blood cell, it's differentiated into an antigen-presenting cell, and we would have what we would call an MHC2 antigen complex there. And it's saying, hey, this is the pathogen, I've broken it into bits, you guys need to do something with it. You need to get some sort of immune response to degrade this pathogen. This is the start of what we're going to call this humoral response. This is also called, and you might hear it both ways, these kind of go back and forth, exogenous antigen presentation. Just think of it as humoral. We're taking an exogenous antigen, breaking it into bits. Now we have this presentation. Everyone good so far with this part? So now the next process takes place. This guy interacts with a second type of immune cell called a T helper cell. This T helper cell has its own immune receptor that interacts. We call this the T cell receptor. Or TCR for short. It's always interacting with that MHC class II in antigen. It has a secondary receptor, co-receptor called CD4. And CD4 might sound familiar to you because when we talked about HIV infection, that's the co-receptor that HIV basically uses to infect white blood cells and T helper cells. So we have this interaction going on between the T cell receptor of the T helper, CD4, this MHC2 antigen complex. Now it's this T helper which does a few different things. But the big thing is it basically produces a crap load of cytokines or signaling molecules. These are specific to B cells. So those cytokines from the T helper activate a B cell. That B cell then differentiates into a plasma cell, and that plasma produces the antibodies, which I now call AB because I'm lazy and don't like to write it out. So the overall process here is to produce antibodies at the end. That's the first one, the humoral system. Also known as exogenous response. So, when you bump into a pathogen, when we get to this part right here, not only one type of antigen is being produced. So for any one given pathogen, so if we start with one pathogen, it is broken down into many, many thousands of antigens. Each one is being displayed on the outside. That means your serum has what we call polyclonal antibody mixtures. That just means it has a mix of all the antibodies towards all of the antigens that you've bumped into. So for E. coli, you might have 5,000 different antibodies that are produced towards 5,000 different antigens just from E. coli. Of course, it's not that many, but just to exaggerate. Now, this is a crazy process in that this just doesn't produce a whole bunch of antibodies willy-nilly and think, oh, maybe 1 out of 1,000 I produce is going to work. We have specific processes taking place so that when that B cell gets activated, it shuffles its genes with purpose. Pretty crazy. It tries to find the right gene combination to make an antibody to bind your antigen. So that's where we're going to talk or start talking about next, is what these antibodies look like. And they're pretty crazy little things. So our antibodies are basically composed of 4 protein chains. Too short, too long. Sometimes these are called heavy and light chains, depending on how old you are and which text you're looking at. And they kind of form this Y. So our heavy chains are these long protein chains as such. Our 2 short chains are connected to the outside and underneath that arm. And then all of these 4 are held together by disulfide bonds to keep its integrity. At one side of the antibody, we have what we call the FC region. This is because it's constant. The genes never change for this region. They're pretty straightforward. On the other side, we have the antigen binding, also known as the Fab region. And we have 2 of them on each antibody, 1 for each antibody arm. These are basically where we see it binding our antigen. And so we can produce a few different classes of these bad boys depending on their structure and their long chains, specifically their heavy chains. The other term for antibodies, the true term, are immunoglobulins. So sometimes you see antibodies called IG or immunoglobulin molecules. They're just synonymous with one another. One's no different than the other. So we have IgG for one class, IgM, IgA, IgD, and IgE for your 5 classes of antibodies. And remember, these guys are specifically made towards your antigen, and we don't have much cross-reactivity, or we try not to. So your IgG is probably the most abundant, about 80% of all of the immunoglobulins in your blood at any given time. It can pass through the placenta, and so it's the major antibody that protects the fetus. You got your IgM, it's the largest. It's actually 5 antibodies connected together, so it's a pentamer. And so if you look at it, they're all kind of connected on the FC region, so on and so forth. So it kind of creates this big round IgM pentamer. First produced to infection. And it plays a large role in clearing out other pathogens as well. IgA, also known as serum Ig. So basically, we find it in serum, mucus secretions, specifically in mother's milk. So this is basically protecting your newborn until their humoral system is up and going and they can combat things on their own. So it doesn't look like a lot of you have had kids in there. I know a couple of you have. But right off the bat, right when you give birth, of course, you don't produce milk. What do you produce? Colostrum, right? All that is just a big syrupy mixture of IgA. Proteins, fats, sugars. And so this is kind of where we see that first beginnings of normal flora being passed from the mother, and at the same time we're trying to protect them from any sort of humoral organism. Now IgD we see in really low concentrations. It in itself is kind of acts like a cytokine. So I call it cytokine-like. And it interacts to help flush out and get rid of other pathogens. It doesn't really act like all the other IGs that we know about. And then the last one is IgE. Also, we see it in low concentrations, but specifically, it interacts with mast cells and is one of the main propagators for what we call the immediate allergic response. So it's basically interacting with those allergens and creating what we call an allergic response. So with this, we can also talk about these things called adjuvants. And these are external, additional things that we can give someone to help with immune responses. So we think that is the ability to sharpen immune responses. And they do things like increase antibody production, we can use them when we do toxin immunizations or vaccinations. They help to make the antigen insoluble. And when it's insoluble, we get a stronger response. And then, like I said, it can manipulate. So we can add various compounds that allow us to switch from one response to another. Good example, once you get an HIV, right off the bat you get large loads of virus in your blood. Your immune system recognizes it, does this great exogenous presentation of that virus in your blood, You produce all these antibodies, and very quickly the virus goes intracellular. So if you can trick the immune response into producing a stronger cellular response to begin with, it helps. So these can be different types of things. So they can be biological. So these can be things like proteins, nucleic acids, etc. But these aren't used very often. Usually it's the non-biologicals. And a lot of these have gotten a bad rep, of course, with this whole autism thing, that these are part of the reasons why kids get autism. And they're not, but it's always good to debate the process. So a lot of these natural, non-biological adjuvants come from the first 2 or 3 columns of the periodic table of soft earth metals, so aluminum hydroxides, aluminum phosphates, are great examples of this. But there are a whole bunch of other ones that they have developed out there as well. So the last thing I'm going to talk about today, and then we'll come back and revisit it on Wednesday, because it's kind of a complex thing, is that up to this point, the humoral response is not about clearing the pathogen. So the antibodies and everything produced by the humoral response do nothing to get rid of the antigen themselves. They have no antimicrobial properties. So rather, the key here is to tell the difference between self and non-self, and then of course presentation. And so what we get out of this self-non-self presentation is an antibody specific to the pathogen, and more specifically the antigens of that pathogen. So other systems can use it as a self-non-self property. So for your humoral response, it utilizes other clearance systems. And one of these systems is called the complement system. And I'll just kind of leave you with a little bit of the complement system before we head to lab. It is a series of 20 plus proteins that get together to basically destroy pathogens. Has 2 different pathways for activation. The classical pathway is an Ig-mediated pathway. So basically, IgG recognizes the pathogen, whistles over the complement system and says, hey, you guys need to kill this thing right here. The second way that we get is the alternative. The alternative pathway specifically recognizes microbial carbohydrates like mannose. It does not use any sort of IgG. And this is kind of a cascade type effect. So think of blood clotting and the cascade of clotting factors that creates the blood clot. We pretty much get the same thing happening. Here's my E. coli or bacteria. I get my IgG recognizing it, and it binds to the bacteria. It's that Fc portion that recruits the first complement proteins. These proteins are called the recognition proteins. And they start to accumulate and grab hold of this and then start to form on the outside of the pathogen. And there's a series of, I think, 10 or 12 proteins that are part of this. The second thing that we get is a cascade of assembly proteins coming down. And then eventually, we end up getting this MAC formation, this membrane attack complex. This is a set of about another 10 proteins that interact together. And what they end up doing is they end up forming pores or holes in the membrane of the E. coli. So they'll end up making all these pores in the membrane, these MAC proteins. And in doing so, this thing just blebs open, lyses, and is destroyed. And we'll leave it there for today. We'll go ahead and interact with our lab stuff here in about 10 minutes once you guys get down there. And like I said, 2 big lab nights and we'll be done. Thank God.